welcome to our first in-person / hybrid event. We're doing a little experiment. Another experiment with the microphone and getting close enough to it. You got me okay over there, Tom? Okay, we're also doing this to audience out on Zoom. So again, experiment this time around. We're hoping this is going to work out, and we'll probably do these in the future if they do. A couple of housekeeping things. It's been a long time, so if you could remember to drop off your name tags in the basket as you leave the building. We'll be recycling those and using them for our next event that's coming up, and I'll talk about those later on. A little shout out. We, as usual, extended invitations to other alumni clubs here on the Cape. So we've got 3 members of the Cornell Club and their guests, 3 folks from Harvard and their guests. We've got 7 MIT folks, including 1 unaffiliated community member who we will be twisting his arm to join our organization. And we are being recorded. And whatever cleanup things do I have here? Yeah, so that's it for our cleanup things. I'll come back at the end of this and talk about our upcoming. We've got 3 events scheduled in the future coming up. Alex, you're all going to be right here. And I'll do a very, very brief introduction to our speaker this afternoon. You've seen the biography, on the website. Hopefully you can recall some of her background. Wanted to mention that although she's not an MIT alumnus, she did spend some time at MIT doing some work on a project of hers in a lab working with a scanning electron microscope, and apparently got in a little bit of trouble for playing the music too loud in the lab. So without any further ado, I want to introduce the director. Is it director of Center for Coastal Studies in Provincetown, Sarah Oktay. Thank you. Again, we're out of practice here.
Oh, yeah, I was up earlier, but I'm. Switch on. Oh, I meant temporary
switch.
Ah.
Always one more switch. It's on its way. That takes a while. No, about 45.
Okay, I'm going to make sure everyone can hear me. Is everyone on Zoom okay as far as hearing me? Hopefully not.
You won't hear back from them.
Okay, yeah, because you've muted them all. Good for you, Tom. Thanks.
They know they can chat. They can chat.
Yep.
So they know that, and we'll answer questions.
All right. Can everyone see that all right? Yeah, some twisting of.
You can do a share screen from the room.
Yes, I will do a share screen from Zoom. Thank you. Actually, I'm going to end this temporarily. Hang on. Just got to share the screen. Yep, to Zoom shortly. I absolutely am doing that. I hear that. Too many icons. I do have quite a few icons. I have very good hearing. Making fun of my icons. I hear you. So I'm getting there. Share screen. Here we go. Blah, blah, blah. Okay. So I'm going to minimize up here. Everyone can see here. I've got to move a couple of things around so I can actually operate this. All right, that should look okay on Zoom, I hope. Okay, and everyone in the room can see. Just did a talk the other day where we were having a ton of people on Zoom and a lot of people in the room. And we had this fantastic speaker named Philip Hoare, who was giving a talk about an artist and whale research, and everyone in the room could hear him, but couldn't see his slides as well. Everyone online could see the slides fantastically and could not quite hear him. So as long as you keep, everyone on the same playing field, then I think it's fair game. So I want to thank you all so much for having me here today. I just became the executive director for the Center for Coastal Studies a year ago in January of 2022. I'm a chemical oceanographer. I have a PhD in chemical oceanography, studied at Texas A&M University, and thankfully studied chemical oceanography, biooceanography, geo, and physical oceanography, which is great because I have to use all four of those every day in my job. And I do have the best job in the world. I run a research lab with 46 scientists in 12 departments there in Provincetown. I get basically paid to see some of the most endangered and beautiful animals on the planet, and every day try to do something to make this a better place. So, but thank you again for having me, and I'll launch right into this. It's a lot of slides, but I'll go through it pretty quickly. Feel free to interrupt me as I'm going. Hopefully Tom or Mike or someone's watching the Zoom. I might not see chat questions, but I don't mind being interrupted, and I'll be able to stick around for some time afterwards. If you all ever want to get a group and together and come down to P-Town or up to P-Town or over, however you want to say it, to P-Town for a tour, I'd be happy to arrange a tour of our labs. So we are a little tiny, mighty marine lab at the tip of the Cape. You can think of us really like a mini Woods Hole. We were started in 1976 with 3 friends that were working on the dolphin whale fleet boats, 3 scientists and biologists named Dr. Graham Geis, Stormy Mayo, and Dr. Barbara Schuler Mayo decided to get together, combine all their interests, and start doing marine science in Provincetown. They were meeting in a little tiny attic apartment for a while, and then they were in the basement of the Chamber of Commerce, and we bounced around all over Provincetown from small area to small area. Now we're in a 12,000 square foot building. We have close to 47, 48 scientists, about over 100 adjunct and citizen scientists, volunteers, interns, a $6 million budget, and a lot going on, as you're going to find out. We have a lot of partners, and I wanted to introduce this slide here so that even though we're located there in Provincetown, we work closely with the Pleasant Bay Community Boating, with our colleagues at the New England Aquarium, at Woods Hole, at Northeastern, at UMass Boston. We have a very strong connection with UMass Boston, and also with all the different groups here on the Cape and Islands, Buzzards Bay, Friends of Herring River. There are so many people that care about the environment that live up and down the Cape, and we work with all of them. 2 big partners for us are Stellwagen Bank National Marine Sanctuary is a very close friend and partner of ours. We serve on their advisory council and do a lot of research in Stellwagen Bank, and the Cape Cod National Seashore. To show you how close of a partner we are, we actually took all the water quality equipment from the Cape Cod National Seashore and took over all their water quality measurements. So we do water quality for them. Here's a list of all our departments. We'll have a little quiz later on so that you can quickly memorize it. This group can actually memorize all of this, so I'm not worried. But we do everything from seal research to shark research to whales, and we'll go through each of them quickly but thoroughly. So we're most famous for our right whale research, and the North Atlantic right whales all come here to Cape Cod. This is a population that is endemic to Cape Cod in the summertime. We're a very important nursery area. So 85% of the world's North Atlantic right whales come to Cape Cod Bay and Massachusetts Bay. Yes.
The Zoom is seeing your presentation, but not your slides advanced. So you shared your PowerPoint, but not the PowerPoint show, not the presentation part of it.
Okay, let me try it again. Then let me stop share screen. Now I have. Does that work? I'm going to advance. Thank you so much for keeping an eye on that. So they're seeing that change. Okay, perfect. Thanks. Yeah, there's too many screens and I appreciate that. So this is, oh, and it's jumping ahead, always does here. So here's a North Atlantic right whale. You might not have seen this because there's only 340 of these. They're here from December till about May 15th. So if you go out on a whale watch cruise, you typically are not going to see right whales. You're typically going to see humpbacks, fins, sei whales. These are incredibly endangered on the red list for endangered creature, probably one of the most endangered creatures on the planet. These are baleen whales. You can see the top of them. Hopefully you can see maybe my little cursor, maybe you cannot. But you can see on the top of those, that white area is an area that is on top, called a callosity. That is actually infiltrated by a type of sea lice to turn white like that. And that is the fingerprint for a right whale. So we identify all the right whales based on that pattern on their head or bonnet. For humpbacks, you identify humpback whales based on their flukes. Their flukes are their signature for humpback whales and fin whales. But for right whales like these guys, we're identifying them based on those little white bumpy parts on the top of their. So the way we do that is we have eyes in the sky. We have the Cessna 337 Skymaster up almost every day between December and May, and we have very highly trained aerial observers that are flying around in circles at 1,000 feet, taking 10,000 photos. Typically every year we have 35,000 photos. So we literally take a photo of each and every right whale from the top. And then we have another team in a boat on the bottom on the water that's taking photos from the left and the right side of each right whale. So we are literally photographing each and every whale to identify them. We're looking to see if they're entangled. We're looking to see if they've got a baby with them. Right now we've got 12 calves that we have observed that have been born this year. So here's what it looks like from the air. This is one of our aerial photos. Anytime you see research done on right whales or articles, typically you're looking at our photos. This is hot off the press. This is from January 8th, and this is number 3391 skim feeding, which is just a highfalutin word for saying feeding at the surface. And it is eating a whole lot of copepods, which I'll talk about here in a second. This is a picture from last year of a right whale, and you can see those bumpy parts. And I included this because it's a great way to see these little warts on top of its head. And those are what gets colonized with that amphipod, that sea lice that we then use to identify it. When you see a V-shaped pattern, or almost looks like a heart for Valentine's Day, that is a right whale blow. So a humpback blow is a single shooting thing. A right whale has a V-shaped pattern. In case you're out and about. This is what, and it's not the world's clearest picture, but this is what our track lines look like. So we fly out in very methodical lines up and down, looking for these right whales and back and forth throughout Cape Cod Bay. We identify harbor seals. We can identify porpoises. I think we had 170 porpoises the other day. Some days we might have 200 whales out there. In addition to seeing all these whales from the sky and being out on the water, we're on the water in a research boat called the Shearwater, and we're collecting phytoplankton grabs. On the right here, you can see this phytoplankton grab, this big pink thing. That is all phytoplankton and zooplankton. We're looking at something called a copepod, and that's what this big orange thing is. Here is a copepod. And that is like a little baby shrimp, kind of like a krill, even smaller. And that's the primary food source for right whales. They're not eating fish. They're eating little teeny tiny zooplankton that you really have to identify under a microscope. They're eating billions of these animals every moment. So we are dragging behind there and identifying that. And that's one thing we do differently than other groups around the world is we actually get a full habitat survey and a suite of all of the animals that they're eating and how that's changing throughout the season. So when you come up to the lab, I can actually show you, this was what the suite of copepods were on February versus March versus April. As the season goes on, we get more copepods called Finmarchicus and Calanus that have more oil in their bodies and they're more nutritious. So they can eat less as the season goes on. Currently, we're also doing some experimental work using artificial intelligence and satellites. So we can't be everywhere. I don't have a team, I don't have a whole army of planes all over the East Coast looking for these. So we're starting to use a variety of different satellites to do imaging of these right whales, especially when they're not here eating. And our observers are so well versed in whale watching and in understanding what each whale is that we can actually identify the whales based on satellite images. So this one in the upper left-hand corner has a little horizontal line and that's ruffian. We know that that's that whale based on the signature on the back of their head. But we can also see the flukes, so we can also identify both species and sometimes individuals. It's been difficult training computers to actually do AI and do image characterization with this. We're working on that. It's easier with people than it is with whales, but we're making some progress. Other cutting edge thing that we're doing is using stable isotopes like carbon, oxygen, nitrogen to recreate their life history and where they've been feeding when they're not here. We take their baleen, which is just like our teeth, it's growing out from their gum line. And typically this much baleen here is maybe 8 or 9 years. We've gone down to the Smithsonian and collected baleen from the Smithsonian, and we're doing stable isotopic measurements throughout that whole length of baleen every centimeter or so, so we can recreate their life history. Stable isotopes in the ocean change based on temperature, based on where they're eating on the food chain for nitrogen, and based on how close they are to land. So it's a fingerprint using these multiple isotopes that we can use for just a piece of baleen, literally sitting in a drawer, that can tell us where that right whale lived during its life. We've also got a marine fisheries program, and I can share this whole PowerPoint if you all want, because all of these links are hot links and go to the entire program. And I'll tell you a little bit more about that in a second. One thing that's important with the North Atlantic right whale is right now there's only 340 individuals. This year we had 12 calves that were born. Last year it was estimated 20 died. Obviously, if any of you have taken population biology, we need at least 20 to be born to keep the population stable. If we're losing 8 or more each year, then that population is going to keep declining. I hate to say it, but there is a good chance that your grandkids or your grandkids' will have not heard of right whales. They definitely are on the brink. They're still viable. They're not reproductively extinct, but it's a serious issue. And this is what the calves look like. Pretty cute. We'll jump ahead to our fisheries research. We have a huge fisheries team. This is Owen Nichols. He's done 27 years of fisheries work here. We work with shellfish populations, and we hire a lot of fishermen. So we hire fishermen to work with us to do work with shellfish, with pelagic fish, which are fish in the water column, to provide them jobs in addition to just fishing. One of our biggest research areas is looking at aquaculture, both for water quality and for making a living, growing oysters. We've overfished a lot of our oysters and mussels and other fish here. And we work with fishermen to develop aquaculture leases that we can then keep an eye on as far as diseases and other things that might be affected. Squid is a big thing that we research, especially in Nantucket Sound. Almost everything eats squid. So squid are pretty much like, I guess, the antelope of the Nantucket Sound. Any of the larger fish are going to eat squid. So they're pretty cute. This is the long-fin squid right here. We did extensive surveys for age classes and population studies. If the squids start dying, and it could be from anything, pollution, vehicle traffic, who knows, changes in sea level rise, in sea surface temperature, we want to know right away if the squid population is suffering, because everything else is going to suffer in relationship. We also do a lot of bycatch studies looking at, this is a gill net on the right. It's kind of a little difficult to see, but we have a whole video. You can probably see the sharks there in the water next to that gill net. So we're putting these, fishermen put gill nets down on the seafloor. They're big nets that they leave for a day, a week, maybe 2 weeks. So fish get stuck in this netting, and then every seal and shark in the area goes, wow, this is a buffet of fish. I'm going to come check it out. And so so even sea turtles will do this. They'll go check out the nets and often get entangled and die. So not only do fishermen lose their nets and fish in there, but they end up with bycatch. And in this case, the bycatch, which is catch you weren't aiming for, is going to be seals and sharks. Sharks might survive because they don't have to get to the surface to breathe, but seals will definitely die. So we've been putting cameras on the gill nets to try to establish what kind of creatures are approaching them, how quickly they approach, how can we help develop policies to reduce that bycatch. Kind of depressing. Not everything I do is fun, I have to say, but everything we do does make a difference. Everything we do, we provide the data immediately for policy changes. Another program we have is our seal program. Been operating this for 20 years. If you all have been following the gray seal issue, they've been rebounding. They were locally extirpated in Massachusetts and all of the United States as late as the late 60s, early 70s. Once the Marine Mammal Protection Act went into effect in 1972, the population started rebounding because we stopped killing them. And it's a very common area for seals to haul out because we have all of these great shoals and lots of feeding areas. If you have a lot of seals, you'll eventually have sharks, which will be the next thing I'll talk about. But this is a success story, similar to things like the bald eagle. So we monitor all the seals. Sometimes we actually monitor them with a little attachment like this, where we're tracking the seals. It's not easy to put one of these things on a seal, and they often will shed it, but it does really help. Sometimes we'll even have a camera on it. Gray seals are adorable. They're actually very different than harbor seals. And when you're out and about, you're typically looking at these giant horsehead gray seals. We're starting to do environmental DNA with gray seals. So do you see all of these guys are green on the back? They normally shouldn't be. That is basically moss growing on the back of the seals, and that's related to climate change and warmer waters. You also can see a whole lot more moss on this water, on the shoal, I mean, and you'll see sea lettuce. Those are both indicators of excessively warm water. And so these greenies, we're wondering how they behave, how they swim. Does this make them easier to find for sharks? Does this make them less likely to have children? Who knows? So this is a new phenomenon, and we're using environmental DNA to actually determine how many seals are using haul out sites. How many people in the room have heard of environmental DNA? One. Okay, that's one more than most groups. Okay, one and a half, two. So environmental DNA, this is a very cutting edge thing. This is literally going out and taking a cup full of water, running a series of DNA samples on it, looking at what it links to genomically. You know, we have all of these full genomes of many different creatures. So we are literally looking for different pieces of DNA that are in the water that are linked to both the phytoplankton, zooplankton, large creatures, us, are all going to be in the soup, all of the things and cells that we shed. And so this picture on the right shows all the things that an environmental DNA a sensor might be monitoring. So you could keep track of where whales are, sharks. You would look for things that have large differences in their genomic signature. It's still a little bit of hand waving, still a lot of assumptions, but it is helping us identify areas where certain creatures are using them. You could imagine for salmon, for instance, if you're looking at rivers, different salmon go to different rivers, you could use eDNA to actually distinguish which rivers are most used by different types of salmon. So we're using environmental DNA and looking for the strength of DNA for seals to see where seals are hanging out. We've got a shark program. If you go out to the museum, the Atlantic White Shark Conservancy Museum in Provincetown, there on McMillan Wharf, you'll see a big exhibit of our shark research. Where there are seals, there are sharks. On average, there are anywhere from 300 to 600 white sharks off the shores of Cape Cod. About half of them are tagged by folks like Greg Scommel, who works with the Division of Marine Fisheries, and other groups like OSearch. So what we do is we install acoustic sensors on the seafloor to look at fine scale measurements of these sharks. So half these sharks have pingers. They're sending out a signal. They don't know that they're doing that, but they are. And so there's several groups that have put hydrophones in a large area to collect signals. What we do is put a whole bunch of hydrophones in a small area to get very fine-tuned behavioral information on these sharks. So this is our shark guy, Brian Laguerre, putting maybe 40 or 50 different arrays. So we'll have an array this tight, and you can see all the way up and down the Cape, you know, the side of Cape Cod. our array all along there. Some of these yellow dots are actually the state's array, and this inset shows you what we do for our fine scale array. Why do we want to do it in such fine scale? Well, we really want to know exactly what the sharks are doing. So when you use the Sharktivity app, you're getting pings and information from this large array, which is helpful, right? If you're at Nauset Beach and you want to go into the water, I would recommend you look at that Sharktivity app and just see who might be around, because at any given time, certainly in July, August, and September, there's probably going to be 300 or 400 sharks within 5-10 miles. There are a lot of them, and most of the time they're not eating us, so there's no need to panic, but they are there. What we're doing is when we do this, the small scale array on the left side here, we're looking at their behavior, and what we found is they actually use the sandbars as cover to hide, and they stay all the way down on the bottom in about 6 feet of water, practically rubbing their belly on the sand on the bottom, sneaking up on the seals. And so we've been able to learn about their behavior and how they actually are able to hunt. Switching gears, I know I'm switching gears a lot. It's 12 departments. I'm going to give you quick highlights of all 12 of them, and then I'll take some questions, so, and I can go back. Humpbacks are a big thing of what we've been doing. We've been doing humpback whale research for 40 years. We keep the catalog for humpback whales for the North Atlantic with over 3,700 individuals. We use genetics, microbiology, mark and recapture, endocrinology. We do work on pregnancy of humpback whales, on their lineage, who birthed who, and how long they've been alive. We have some humpbacks that have been visiting us for 50 years and coming with their calves. So we keep the Gulf of Maine humpback whale catalog, and we're just now building a new genetics lab in order to do humpback whale research. And here on the left is Dr. Juke Robbins. You might have heard her on NPR last week. She is shooting her crossbow, and she's taking a genetic sample, a biopsy of this humpback, which is way out in the distance. So it's not like with you all who are going to stay still and I can actually take a piece of your skin. With a humpback, you actually have to shoot it from a distance with a little tiny fine scalpel that takes a little plug of the skin. You reel that back, and that's what you examine for genetics. If you're lucky and you are able to get a blubber sample, you can start looking for things like cortisol and endocrine. You can see if they're pregnant, what level of stress they're under. So we do 2 different levels of genetic sampling. On the right is our marine animal entanglement response team, and that is the team that often goes out and works with the humpback team. They work together on the same boat, looking for entangled whales and collecting research on the whales that are not entangled. Switching to our marine debris and plastics program. Got a huge marine, in fact, we might be getting a very large grant from NOAA this week on marine debris. We have 200 volunteers that help pick up trash. And not only do we pick up trash, we actually quantify every piece of it. I don't mean we just count each and every cigarette butt. We actually quantify how many pounds of plastic, what types of plastic, how many paper, so that we can have very accurate policy measurements based on the amount of trash we're seeing. We have about 200 volunteers, and on average, we pick up about 50,000 pounds of trash a year. We also have a very involved marine plastics program. The material on your right is actually shock tubing that was used in the Boston ship channel to dredge. So that is actually was forming the fuse for doing some dynamiting at the bottom of the ship channel. When they were dredging this last year, this material washed all the way down here. Typical oceanography tells us exactly how things are transported. No one had ever seen it before. What's helpful is we knew within 5 hours that it arrived here, and we were able to reach the engineers and get them to change their processes. So they were trying to corral all of this material using basically drapes in the water column to try to pick up like you would do for an oil spill. And obviously this material is getting up and over the drapes and under the drapes, so they weren't corralling at all. So this changed exactly how this entire process was. We're currently also doing marine plastics measurements where we're degrading plastics and measuring the spectrum of those so that we can determine what is happening with microplastics. We even go so far as to use our sonar systems to map the seafloor, find marine debris, abandoned gear on the seafloor, which is called ghost gear, bring it up and return it to the fishermen that lost them. So we're cleaning marine debris before it actually washes onto our shores. Microplastics, I hate to say it, but I'm sure everyone in this room has some microplastics in their body, probably some evidence of it in your hair. I mean, it's everywhere. Macro becomes micro as it breaks down in the water column and on our beaches. And we do a lot of measurements to determine what microplastic levels are existing inside animals. We can even, in those little copepods that I showed you earlier, we can even identify microplastics in them, which is a bummer. What's less of a bummer is that we have so much marine debris that we have a cadre of about 25 artists, and we've become marine debris brokers and marine trash brokers to the artists, providing them material to do artistic exhibits like this one at MoMA in New York City. So we have so much leftover lobster line and plastics that a whole bunch of people are using it to make art. Our water quality program is probably the most important to everyone here in the room because a lot of that work is done in this area. We have 150 water quality sampling stations. That's a picture here on the left shows all of our water quality sampling stations, which go all the way up to Plymouth and out onto the island. So we've been sampling water quality of these 150 stations for close to 30 years. And we do everything from nutrient analysis to different types of chlorine, sulfur, ammonia, the whole swath of nutrients for nitrogen and phosphate, looking at all of our fertilizer and septic inputs. We also do monitor for, have in a couple of small projects, contaminants of emerging concern. This would be things like PFAS and neonicotinoids and all the other things that you've been hearing in the news lately. These are different contaminants that are showing up from pharmaceuticals and from pesticides and from all kinds of things. And you can see that we have data on our website for each of these areas here. So you can do a compare and contrast as to where you want to move or not move. You can find all our data. We provide all our data for free, really, to anyone who asks, but it is hosted and served up on the Cape Cod Bay Monitor.org, on our website, on the Association to Preserve Cape Cod, and on the Cape Cod Water Protection Collaborative. So if you go to the Cape Cod Commission, all of our data is here. If you see on this lower left, that's exactly what that interactive portal is going to look like. You can click on one of those dots and get any kind of information you want, from something collected last week to something collected 10 years ago. On the right is the State of the Waters report that just came out from APCC and the Cape Cod Commission. Unfortunately, about 85 to 90% of our embayments are severely impacted. See all those little red dots? Red is not good. And so we have, I think it's 82 out of 90 embayments are impacted. And you can get that State of the Waters report on our website and on Cape Cod Commission and learn more about your estuary. Almost all of our research is typically saltwater. This is what one of these spots looks like. Here's one that's in between Martha's Vineyard and Nantucket. You can click on this and you're going to get an idea of temperature, water quality, changes in chlorophyll and in nitrate and phosphate. So you can see how eutrophied it may or may not be. Here's all, I'm not going to discuss all of these, but we do a lot. So if you go to our website, you can tool around, look at our eelgrass studies, look at some of the oyster reef restoration studies. We've got a lot of cool little things there on our website. And I already told you about that. So we're going to go on to the MARE team. This is the ones that get in the news all the time. So basically, we operate a SWAT team for disentangling whales, and our SWAT team is the best in the world for this. We actually train teams all over the world for the International Whaling Commission. So last week we were in France training a team. We've got a team coming from Africa next week. So this is what our team does and I'm sorry, this is a disturbing picture. So this is a right whale that's very sick. See how the head area here has a ton of lice? That's because they can't actually get down in the water column. And so those lice are actually overtaking the callosities there. So, and this is a whale that's essentially hogtied with fixed gear. So the biggest problem, remember we were looking at those whales earlier, they've got a big giant open mouth that's about, 10 feet across. Their eyes are way out here. They're going through the water column between here and Maine. During much of the year are up to 1 million lines of fixed gear, traps on the bottom, big line, and then a buoy on top. Some of those are mooring lines. The majority of them are crab fishing and lobster fishing lines, various fixed gear. So they just plow right through it. I mean, they don't even see it. And as soon as I tell people, I said, the way this feels is basically like when you're hiking and you walk through a spider web. First thing you do is start twisting and turning, trying to get that spider web off you. That's exactly what they do when they hit one of these things. They start twisting and turning and eventually hogtie themselves. So if we're lucky, either a fisherman, a dolphin watch, the Dolphin Fleet Whale Watch boat, any kind of a steamer captain, somebody sees this, or we see it from our boats or planes, and they call it in, and we have a team of people that have to be within 20 minutes of the lab, can drop what they're doing, say goodbye to their families on Thanksgiving, whatever it is, and get all the way up to our lab immediately, get onto the IBIS, that fast boat, and get out there and disentangle us. It takes years and years to be a high quality disentangler. It's not easy work. There's a couple of movies that are coming out or have already been out that are about our disentanglement team, and you can go to YouTube and watch some of it. It's very chaotic, but it actually is, well-trained. They're well-trained for all that chaos. So here they are on the IBIS, which is this high-speed response vehicle. Obviously, we can't respond at night. We never get into the water. If you get in the water, you're going to die. Typically, these whales don't know you're trying to help them. In fact, one person died up in Canada. One of the people we had trained died on board a boat just from a whale hitting it with their fluke, hitting the person. But we go out on these little Zodiacs, and we use the same techniques that whalers used. We're using harpoons, we're using kegging, we're using grappling hooks, all of this very kind of, old world type techniques to try to grab onto the line and cut it off the whales. Jumping into marine geology. One person in here was asking about Graham Geiss's book here. This is in the lower left-hand corner. We have a huge marine geology team of 10 people that have been doing marine geology research for 46 years. Much of what we know about the Cape, certainly the Chatham area, but all of Cape Cod comes from Graham Geiss in our research. We do seafloor mapping studies, we do benthic habitat studies, and storm tide pathways. Here is an example of what mapping a harbor would look like and differences in depth. Here's Katie Castano, our new seafloor mapping lead, and she is doing the restoration work for Duck Harbor, measuring what's happening with sediment changes when they remove a dike that's been in the water for over 100 years and restoring this area. So we're doing measurements on fine scale sediment recovery to make sure it's not covering up the oyster beds and see what's happening with overwash. I'm not going to read this whole thing, but this basically says we know what we're doing. We're using a lot of high scan sonar and very intricate imaging software, in addition to positioning software that tells us if I want to tell you how tall something is to the millimeter, I have to know where I am. And so that actually takes a lot of computing power and a lot of Trimble GPSing. There's a lot involved. And so that's what this talks about. When you have all that information, you can tell where something is to the millimeter. This is what our boat crews look like with their side scan sonars, with backscatter images. They're out there mapping the seafloor. We can tell if to a beer can. I actually could tell down to a probably snuff can, probably not to an earring. If you lost your earrings, I don't think we can help you, but we could, if it was a bracelet, we probably could. So here's what some of the mapping around the Boston Harbor Islands looks like. So we do a lot of work for the park system, looking at the various islands, looking at stuff for Boston Harbor. We've mapped almost all of the bayments up and down through the Cape. We use a lot of this is stitched together imagery. It looks like it's all photographs, but it's actually sonar images. We've also got a large app that if you live in any of these areas, as I slowly bring this up, we do something called storm tide pathways, and we've mapped all of these areas and are finishing this portion right here in Cape Cod, so right between Chatham out toward Woods Hole. So you go to stormtides.org and you can get information on exactly what streets will flood when at different levels of sea level rise in different surge areas. This is sometimes, nice weather flooding just on spring tides. Sometimes it's associated with major nor'easters. So we have apps that say street to street, and many times these places flood from the back end. They'll come through one area, one low point, and then come in through the back way. We've got a drone guy, full-time drone guy. When I talk to high school students, I'm like, how many people like drones? And every arm in the room goes up. If you don't like whales, you definitely like drones. And we actually hire someone year-round as a drone person, and they're out mapping the Cape Cod National Seashore. They're mapping the harbors, shallow areas, constructing these maps that show exactly what's happening over time. Some really cool imaging. We just got this new piece of equipment that was designed at Woods Hole. It's called a jet yak. It is basically an instrumented kayak with about $50,000 worth of equipment on it for doing near shore side scan sonar research. It was typically used in glaciated areas with glaciers where you couldn't get close to a glacier because it's not safe. Glaciers, when they melt, can sink your boat. So now we're using this here in our embayments. Couple more slides and we'll be all done. Talking about benthic creatures. This adorable thing on the left is a very, very young horseshoe crab. We do tons of benthic studies. So not only are we looking at the water column and we're looking at the big giant whales that we see, we're going down to the sediment and taking sediment samples and doing full community suite analysis using our imagery software and about 200 volunteers to count all of the creatures that live in the sediment that we sample. We did a full reconstruction of Pleasant Bay, taking about 25 different samples over 2 or 3 seasons to see what parts of Pleasant Bay are the most healthy. A healthy benthic layer, bottom layer, and a healthy water column tells us the exact health of a embayment or an estuary. If you take a sediment sample like you could in Boston Harbor in the 70s, and all you find is blue mussels, that is not a healthy system. You're going to want to find 25 or 30 or 40 different creatures. In our case, we found a total of 400 different creatures and 400,000 individuals. Took us 5 years and about 100 volunteers to count all of these. But we have the most thoroughly studied parts of the benthos, more so than many other areas. And this is what these adorable things look like. And this is what a day picking out these adorable things under a microscope look like with all our volunteers. We're using side scan sonar. I'm sorry this got blurred out on the left, but we're actually using side scan sonar to look at squid eggs. So on the right here are a bunch of squid eggs that we're using side scan sonar to monitor so that we can look at squid populations. And last but not least, we've got an education program. We have one poor guy named Jesse that tells everybody about everything we're doing. Mainly all of our K through 12 students up and down the Cape. We go to the 5th, 7th, and 8th grade, talk to them about water quality. They conduct the same experiments year over year. So we actually have 10-year-long data on water quality for some of these schools. And he goes out and to all the high schools too and talks to them about right whales, about seals. We have in Provincetown not only an exhibit, we also have pop-up tents on the beaches where we talk to you about seals and sharks. And we have something called Seascape that is down on Commercial Street, all education-based. Here's a few of our vessels. I know a lot of you folks are plane and boat people, so I decided to show you a variety of our vessels. We have 5 of them that we use all the time. We also have a kayak and a canoe that we use for research that we call the Sea Wolf, which I think is kind of funny. This is all of our tools that we use. We do global training through the International Whaling Commission, and this is all of our disentanglement tools to the right here. And you can see how much they look like harpoons. They're basically harpoons designed so the pointy bit goes backwards instead of frontwards. And that's how we help cut all of the lines off of whales. We actually make those tools locally and we provide them at cost to teams all over the country. Here's a picture of spinnaker. Spinnaker is our right whale. I'm sorry, not right whale, humpback, 11-year-old humpback whale. And when you come to the offices there in Provincetown, we're open 9 to 4. And unfortunately, her skeleton for this young juvenile female whale who was just reaching puberty is up in our rafters instead of swimming around like she should be. She was entangled multiple times. We disentangled her 3 times, but it was not enough. So this little piece of gill netting is what did her in, and this is what you see when you come visit us. This is a piece that we couldn't get out, almost like a piece of floss that basically killed her. Anyway, it's a very powerful exhibit. You're welcome anytime. It's free. I'm usually there in the office. We've got some exhibits that talk about her life. We've got an open house on May 20th. And we also have that exhibit at the White Shark Conservancy Museum. Here's a full-blown image of spinnaker. One thing you can maybe see in this picture is the two little pelvis bones. A lot of kids and people don't think about how closely related whales are to us. And a lot of them will still have a pelvic bone that will be right below the tip of the tail. You can see, look at our phalanges and really, get a feeling for how close these animals are to us. We're regional, local, and global. We do research all over the world. I didn't have a whole lot of slides for our other work, but we work in the Bahamas and Polynesia, all over the place. And that's it. It's good. Thank you. And I see that it looks like there's a lot in the chat, and I'll take any kind of questions. I know it's a lot and they're really short, but I was trying to give you an idea of the breadth and width of everything we do. Yeah.
On the side of water monitoring sites.
Yeah.
It seemed like there were none on the Buzzard Bay side or the National Seashore side. Are other organizations responsible for those? Or?
Typically, we've been reoccupying the same sites we don't have that many offshore here that are permanently occupied. We'll tend to do a whole cruise instead of having a site we reacquire. And then we do have some folks in Buzzards Bay. The Buzzards Bay Water Coalition does a ton of work out there. So they provide us with their data. We provide them with ours. So we assist them. They act as like a farm team, especially where Coit Bay does a lot of work with us, as does Woods Hole. So most of those areas, I can flip back there and we add some sites and subtract some sites. Okay, now making people nauseous, but I'm going to blame that one on Mark Borelli. Almost there. Okay, that's just for the contaminants of special concern. We only did a few sites. So this one on the left shows all the active sites. All those little blue dots are our active sites. I don't know if you can see that, but it's quite a few. But yeah, you're correct. There's none up in here in Buzzards Bay. And then if we go back a couple more, we don't show any over here in Buzzards Bay. So those are all occupied by other folks that are doing that research. And try to see, yep, there's nothing here too. So we go all the way out here. I think 3 bases as far as we get out. We do go all the way up to Plymouth, but I think there's a lot of folks from Woods Hole doing this. We have done one-offs in Buzzards Bay. That's a really good question. Yes, you're next, and I'll come back.
Are you looking at the potential effects of climate change on water levels and tides?
Yes. Yes, that's a big part of what Mark does. We tie in with the USGS. We're out there taking water levels every day. I'm actually doing a talk at Nappies next week just on climate change, warming sea surface temperatures, and biological effects. So we do a lot of, in fact, all of our storm tide pathways data, which was completely accurate up until 2019, already has to be, all those models have to be adjusted because we're still seeing such increases. So we've gone from 1 foot per 100 years to 2 feet per 100 years just in the last 20 years with sea level rise. So we're working, we have, if Mark was here and we were doing just a marine geology talk, it'd go a whole hour just on the different things we're doing. And things are getting flooded that we just had in Provincetown, some unusual flooding on the east end that normally doesn't flood. And what happened were several houses were raised because of FEMA requirements. And so they were lifted up and that became a new storm tide pathway. So something that was blocked before physically by a house that now became a new entrance for water. But water is sneaking up and around in quite a few areas. And we do a lot of work with the Cape Cod National Seashore at Nauset, for instance, looking at what's happening with their parking lot, what's happening with erosion, coastal erosion. We've got some really cool videos of Chatham and Pleasant Bay showing all of these islands becoming islands and then separating and then connecting. And I mean, seeing the dynamics of all the areas based on coastal landforms and then the increase in sea level rise, which is sneaking up. We just had the storm of record in 2018, and that beat out the 1978 giant storm. So our most recent storm record was in 2018, but we had equivalent flooding just 6 weeks ago around Christmas time, almost as bad as the storm of record. So yeah, it's a big part of what we're doing. It's probably what we're most concerned about. It's a great question. So you and then you.
So could you comment on the various processes that are reducing the population of right whales and what can be done to reduce that?
Yes, so there's two main causes for right whale deaths, and they've been happening for years, and that's ship strikes and these entanglements. So when we do necropsies on right whales that have died, most of the time we're going to see blunt force trauma from a large ship hitting them, or giant prop marks from being cut into pieces, or we're going to find entanglement issues. There's a few of them that will die from stress and from, disease, but that's a smaller number of them. We also do a lot of acoustic monitoring to listen for sound and noise. And we're working with a lot of organizations to see what other factors, but those are the two biggies. So when you have to only go 10 miles an hour or less, that's our fault. Because going less than 10 miles per hour allows vessels to see the right whales before they hit them. So a lot of vessels now also have to have an observer on board. And even for research vessels like us, we have to throttle down to 10 knots. So slower is speed kills. And slower is always better for any kind of boats. We've also changed, for a long time, we had a set of acoustic measurements going into Boston Harbor because they really love to feed right where the main shipping channel is into Boston. And so we've actually had them move that shipping approach slightly out of the paths of the humpbacks, but the humpbacks literally are lined up with the primary shipping channels. So it's really critical there to have really good observers looking for whales. So that's one easy thing. The other thing is the entanglement issue is basically, and you'll notice I'm not eating lobster, won't be eating lobster, unless they're hand caught. Using on-demand lobster gear helps. So getting that line, we're not against lobsters, we're against lines. So whether it's a mooring line, a lobster line, a crab line, any of those lines going all the way down to Florida and all the way up to Canada, that's what they're getting entangled with. And those things it takes them about 6 months to die. Some of these whales are literally dragging gear from Virginia to here, or from Nova Scotia down to here. Each different state, Massachusetts actually does the most for protecting right whales. There's no lines allowed into the water until we do 2 days of flights in May and say all the right whales are gone. Then all the lines go in. Right now, there's no lines allowed in the water. So those are the two easiest ways to help fishermen, and it's expensive to go for on-demand. I don't know if you've seen the on-demand fishing for oceanographers. It's very common. So you've got your gear down on the seafloor with an acoustic release, and you just power up to it and hit a button and it opens and your buoys come to the surface and it brings your traps up, instead of having a line sitting there the whole time with the buoy at the top. So then you can just recover that, and that can be connected to the end of like 10 traps. So that's why it's called on-demand fishing or ropeless gear. So that's one thing that we're advocating. It's expensive. They're still working out the kinks, but a lot of those fishermen work with us to try out their gear. So those are the two things. And warming temperatures, basically these right whales, because of climate change, are going further afield to chase their food for the copepods and the humpbacks for their fish. So they're going into areas that aren't protected, because that's where the food's going. It's not like they're going to stop right there and go, oh, this is as far as we're going. So climate change is really a huge factor on this. And so they used to go to the Bay of Fundy, and now they don't go there anymore. They go into the Gulf of St. Lawrence. And if we want to talk about shipping hazards, that's basically like if you lived in the middle of, the country in New York City, and then you decided your dog was going to go down and live in New York City itself. I mean, that's the level of traffic that they're now enduring. So, and that's more of an existential, what are we going to do about climate change that's going to affect all of us? But fortunately, right whales are going to take one for the team before other creatures. It's a good question. Yes.
Why don't right whales have more calves?
We're working on that mainly because they're dealing with entanglement. The last 2 years ago, snow cone was the first right whale to ever have a calf while entangled, literally and unfortunately, both snow cone herself this year or the end of last year probably didn't make it. She still had her entanglement on, even though we tried to disentangle her multiple times. So the entanglements cause their stress hormones to go through the roof because they're spending so much time trying to get out of this. It's affecting sometimes they're wrapped around their rostrum and they can't actually feed. They're dealing with noise, all of our ferries, all of our boats create noise in the water column that stresses them out. They're dealing with being hit by ships. One calf was hit 4 times in the first 3 months. So even the ones that do have calves, but we're seeing the females, we thought we had 90 females of reproductive age. So let's say between 12 and 60 years of age. And now we think we only have 70. And they don't used to have calves every 3 to 4 to 5 years. Some of them still are on that schedule. Others are becoming 5 or 6 years. So it's becoming a longer period of time instead of shorter period of time. So what we really need to do is protect the females and the calves and provide more protection. It's still, we could still dig out of this hole, but it's getting close to, and that's, a super good question. It's true for many other animals. Why aren't they having more babies and more frequently? It's one of the things we're most concerned about. It's a good question. I saw a couple of other hands. Yes, you?
Well, I was interested in what you said about how to solve the entanglement problem. Being MIT people were always trying to solve problems, right? So,
Absolutely.
but the issue is money.
Yep.
And so my question really is, where might the money come from to help the lobstermen implement these solutions? Because it's not going to be for free.
They're already under the, yeah. There's two ways to approach that. The federal government's, there's a couple of, I think Seth Moulton and another senator have put together the Right Whale Protection Act, which provides money actually to fishermen to help pay for these. And I think that's like 13 million. I'm not sure if it's still kind of languishing in committee right now in DC. So providing more money to fishermen to use this. In Galveston, we had the same situation where we had the turtle excluder traps for shrimp fishermen. So when you do shrimp trawls, they would catch sea turtles and drown them. So they developed trap doors so that the sea turtles could get out, but the shrimp didn't get out. Simple engineering. The other thing is demand. When we were all kids, you all remember the dolphin-free tuna, right? And before, you could kill all the dolphins you wanted for Charlie Sunkist tuna. I mean, they didn't care. Once public opinion got out there that all these dolphins were being killed as bycatch for large tuna trawls, then they changed those trawling methods to keep dolphins out of those giant trawls. And now we have, I hope, most people are eating dolphin-free tuna. You remember that on the cans, right? So that's a public PR thing. So if people start insisting on having, when they go out to dinner, lobsters that are right whale free lobsters, that are non, whale killing lobster. And people hand dive for lobsters. There are lobstermen that do fish in ways that are more protective. So providing them that economic incentive, let's say, I would pay $1 or $2 more a pound for lobster if I thought, I wasn't spending $1 million trying to rescue a right whale because of it, certainly, but I would pay more for lobster that was responsibly harvested. And we do that for GMOs. We do that with all kinds of, humane eggs. Look how much we're paying for eggs now, but I'm fine with that because we're having chickens treated better and less polluted waters. So that was all consumer driven. So I think it has to, I don't think that the government has enough money to fix it all from their side, but I think if we approach it from both sides, that we can get there. And there's tons of fishermen and lobster folks that care. It's not like they want to kill. Nobody wants to kill a whale. It's not like they're evil up there going, how many whales can I kill? No, I mean, they're, and it's bad for their gear. Every time their gear gets wrapped up around the whale, they're losing thousands of dollars worth of gear is on its way to Nova Scotia. So it actually helps them. One thing I didn't know, and we're still working on, is the weak. They were using weak rope that broke easier. But one problem with that is if it gets entangled with it, we can't grab that rope. It breaks right where their mouth is. And so we have nothing to grab onto. So one of our captains of our disentanglement team is not a big fan of weak rope because it makes his job actually harder because there's just nowhere to grab. And it still doesn't, the part wrapped around their body stays. It's just all the other pieces. At least they're not dragging, 40 pounds of gear, but that's a great question.
Mhm.
Yeah.
Why are the humpbacks relatively successful compared to the right whale?
Different foods, different feeding mechanisms. So the right whales are skim feeders, they're slow feeders. They stay at the surface. They're very cryptic. They're very quiet. They're kind of sneaky. I actually came upon a right whale up in Boston Harbor in the fog. And I mean, we almost hit it and it just popped out of nowhere. So it's very easy to hit a right whale as far as, and they feed higher in the water column. They tend to feed in areas where there are more lobster lines. The humpbacks also get entangled. Almost all humpbacks have at least 1 or 2 entanglements, but they have fewer ship strikes and their reproductive rate is better. And they've got places to go completely away from humans. So there are places in the world where they go where there's no human impact. The southern right whale, for instance, is happy as fat and happy, and they're doing well because they're nowhere near fishing or shipping lanes. So their normal place to live is away from all of the human impacts. So humpbacks have been taken off the endangered species list. They're still protected, but they've recovered to about 5,000 or 6,000 individuals, maybe 10,000 for this one population that I'm talking about. It's a good question. And humpbacks also eat fish, instead of copepods. So they have different areas and they tend to go deeper. Though you guys did probably see that humpback that swallow that guy. That's because they're eating fish at the seafloor. They literally do just, head to the seafloor and blow air out to scare up sand lance and take a big gulp of them. I have pictures on my Facebook page of, fish jumping out of whales' mouths because there's just so many of them. But that really happened. That guy did actually get gulped. Didn't see it at all. Can you imagine 1 second later, you're trying to catch fish. You've got a guy with flippers and a tank on in your mouth. It was not good for anybody involved. You had a question. We actually have done in a tandem with Salem State University or the Salem State College and with some folks at Woods Hole, we've done a big survey of people to see if they've changed whether they come to the coast. So there's 2 questions there. One, can fewer people go out and swim? Certainly if there is a shark alert, you want to get out of the water. Most people that actually go to Cape Cod to the beach, very few people get out in the water. If you spend a lot of time or get out in water deep enough for sharks to get them. When you think of how many sharks have been there and the fact that we've only had 1 fatality in the last 3 years, and before that it'd be in 100 years, it's actually very rare for someone to be hurt by a shark. But we've noticed tourism actually going up, not down. And you could argue with gray wolves coming back, with bison coming back. This is all risky. These are risky creatures, grizzlies, things like that. So these are all risky creatures that we're allowing to come back. We have to, as humans, adapt what we're doing. But most of the lifeguards, there's still just as many lifeguards out there, just as many people in the water. We're actually seeing benefits to Cape Cod and the islands as far as tourism, number of people visiting, impact on people's cash registers have all been positive. But yeah, you could argue, there's more sharks out there. If you're afraid of getting eaten, I would stay. Yeah, no, it won't be so many. I mean, it's really, they don't want to eat us. It's a huge mistake if a shark grabs a person. It's a completely mistaken identity. 99.999% of the time, they're not going to get us. So I don't really think that it'll affect our grandkids. And I don't think that we would want a world where we would have to go kill all the seals, which I'm not for. And then eventually all the great whites would migrate somewhere else. But I'd rather have my kids see seals and sharks. But it is a higher risk. I just don't think when I talk to my shark guy, when he's on TV, people are like, aren't you afraid? And he's like, no, there's tons of sharks here and nobody even notices they're here. If we didn't tell them, no one would know. So it's actually not like we're all going to die Jaws scenario. It's more than if they were all gone. It's not 0. I'm not saying strap meat to yourself or seal costume and throw yourself out there. But I think I'm not worried for our grandkids.
Does the research on animals?
We're very careful. There is a lot of controversy. There are certain groups, and I'm not going to name them, that do research that we don't approve of and we don't talk about them. That does increase their stress hormones. All the research we do is all with cameras and imaging, not with bringing the animal on board. And like Greg Scommel tags them in the water. He doesn't bring them on board. If you're bringing them on board a research craft, it's very hard to actually keep them wet enough. And you can see their stress, their cortisol just goes through the roof, as it does for us when we go to the doctor and people start slapping equipment on us. It's the same thing. Yeah. So it does increase their stress hormones. We're trying to get better about sampling animals in a least destructive way. And that's one of the reasons we don't tag. Everyone says, why don't you tag all the right whales? Why do you have to worry about using satellites? The tags become infected. Right whales also like to roll around in groups in something called a surface active group, because that's how they make baby right whales. And they tend to rub the tags off of each other, which is kind of like rubbing a piercing off. So we typically don't do any kind of installation of tags because they can cause not only stress issues, but infections sometimes. That makes sense? Okay.
We can
Yep, you're at the
do a
time.
wrap.
Yep.
Just hop over here for 1 second. Whoa.
Oh, and I have a sign-in sheet if anyone wants our newsletter and all kinds of brochures and stickers and other kinds of.
Right for the questions in the chat.
Oh, that's right.
Pardon?
Are there any questions in the chat?
We didn't see any coming in from the chat, and we didn't do a good job of getting the questions over there. So next time around, we're gonna have a secondary microphone to help out the Zoom folks. But there was a small audience this time. It'll be different next time around. We're experimenting here. We're learning. So sir, you snuck away before I had a chance. This is our MIT Club parting gift.
Thank you.
Great.
Thank you.
Yeah. So with that, I just want to, a couple of housekeeping things. Again, if you can return your it's easier. You can return your badges to the bucket on the outside. And then upcoming, we are going to have March 15th, Dan Wolf, CEO of Cape Air, is going to talk about electric aircraft and other avionic topics. April 25th, we're going to have our joint meeting with the Harvard folks. And that's going to be a fellow talking about blockchain technology, crypto, and related issues. And then May, date TBD, we're going to have a tour of the Barnstable wastewater treatment plant, which is one of the largest civil engineering projects on the Cape, I guess, until they build our new bridge. So with that, pardon?
That's not on the case.
Well, yeah, the bridge is sort of straddles the Cape. So yeah, so technically it's on the Cape. And with that, if you have any feedback, you've got emails for Brian and I. We look forward to having some more of these in-person events in the future. Any wrap-up questions, comments? Okay, great. Thanks so much.